Today's deep dive is built around this really groundbreaking 2026 paper from the journal Cell.
Yeah, by Marino and colleagues. It's a massive study.
It really is. And our mission today is to exhaustively unpack how the physical scaffolding of a cell, like the structural beams and roads, is secretly acting as a master genetic controller.
Which is wild because that completely overturns decades of basic biology.
Right. But to understand why this paper is sending such shockwaves through neurobiology, we have to start with a clinical mystery. So I want you, the listener, to imagine a patient who comes into the clinic. They're suffering from severe epilepsy and profound intellectual disability.
And you run an MRI, expected to find a tumor or maybe signs of a stroke.
Exactly. But instead, the scan reveals a condition called periventricular heterotopia, or PH.
And PH is this diagnostic puzzle that has frustrated neurologists forever. When you look at that MRI, you see these dense clumps of neurons stranded deep inside the brain.
Yeah, they're huddled right around the fluid-filled ventricles instead of, migrating all the way out to form that highly folded outer layer of the brain, the cortex.
Right, and because of what we saw in those static scans, the medical community universally classified PH as migration disorder.
Like a traffic jam.
Exactly. The assumption was really simple. We thought the neurons were born normally, but they just got lost on the highway out to the cortex.
That is the textbook explanation. But this new Cell paper completely dismantles that assumption. And to grasp how radical this is, think of a cell as a massive, highly regulated city.
I love this analogy.
Thanks. So the nucleus is City Hall, right? It's heavily guarded, keeping all the DNA blueprints locked away in the vault. And outside City Hall, you have the cytoskeleton.
Which is this vast dynamic network of microtubules and actin filaments.
Yeah, they act as the roads and the steel scaffolding of the city. Now, biology has always operated on this strict zoning law. The road materials stay outside City Hall. The blueprints stay inside.
But today's source material proves that not only are the road materials inside City Hall, but they are actively opening the vaults and rewriting the blueprints.
Which is just insane.
It is. And to prove this, they had to overturn a lot of dogma about how neural stem cells operate. These are the progenitor cells that divide and eventually differentiate into the billions of neurons in a mature brain.
Right.
So to figure out what was really happening in PH, the researchers first had to map the normal protein landscape inside the nucleus of a healthy neural stem cell.
And they did this using subcellular fractionation combined with liquid chromatography, mass spectrometry, or LC-MS, right?
Exactly.
Okay, let's unpack this, because my immediate thought, and probably the skepticism you're feeling right now listening to this, is about contamination.
Oh, absolutely. It's the first thing any peer reviewer would ask.
Right, because if you are mechanically and chemically lysing cells to separate the nucleus from the cytosol, things get violent. The nuclear envelope is fragile. How can we be entirely sure the cytosol, which is packed with cytoskeletal proteins, didn't just, spill into the nuclear fraction during the prep?
Well, the researchers anticipated that exact critique. They didn't just do a standard spin and a centrifuge. They used highly stringent density gradient centrifugation.
Ah, okay.
Yeah, and then they performed rigorous Western blotting controls. They probed for known strictly cytosolic enzymes like GAPDH.
Which shouldn't be in the nucleus at all.
Right.
Yeah.
And they also probed for strictly nuclear markers like histone H3. The nuclear fractions were pristine. There was zero cytosolic leakage.
Wow.
And once they fed those pure nuclear fractions into the mass spectrometer, they measured the mass-to-charge ratio of the ionized peptides to identify exactly which proteins were present.
Which leads to the absolute proteomic shock of this paper. When they analyzed the data, looking at the top 10% most abundant proteins safely locked inside the nucleus, they didn't just find transcription factors.
No, they found massive quantities of cytoskeletal proteins.
Tubulins, actin, intermediate filaments. The structural scaffolding of the cell was sitting right next to the DNA.
And among all of those structural proteins, one specific molecule stood out as the primary focus of this research. And that is MAP1B, or microtubule-associated protein 1B.
And for anyone who studies neurodevelopment, MAP1B is incredibly well known.
Oh yeah, it's famous.
But strictly in a cytosolic context. Its traditional role is binding to the microtubule network to dynamically regulate how a young neuron extends its axons and physically migrates through the brain tissue.
Right, but the LC-MS data, which they brilliantly backed up with confocal fluorescent imaging, by the way, proved MAP1B is highly enriched deep inside the nucleus of neural stem cells.
It's basically living a double life.
It really is.
So, before we look at what it's doing in there, let's just synthesize where we are. We've established 3 major shifts in our understanding so far. First, cytoskeletal proteins don't just exist in the cell body. They are highly abundant inside the nucleus of neural stem cells.
Right.
Second, subcellular proteomics proved this isn't an artifact, it's a deliberate feature. And third, MAP1B is the prime example of a structural protein moonlighting in the nucleus.
Exactly.
So think about that for a second. If you were a structural support beam, what business would you have sitting in the control room?
Well, to answer that, we have to look at the functional assays the team performed. Because it turns out MAP1B acts as a biological double agent.
Okay, tell me more about that.
So in the cytosol, MAP1B promotes differentiation. It interacts with a signaling GTPase called Rho-OA.
To help the stem cell mature into a committed neuron, right?
Exactly. It essentially tells the cell, stop dividing, grow your branches, and get to work.
Wow.
But what's fascinating here is, inside the nucleus, MAP1B does the exact opposite.
Wait, really? The exact opposite?
Yes. Inside the nucleus, it actively promotes stem cell maintenance. It suppresses differentiation, forcing the cell to remain an uncommitted progenitor.
Okay, that is wild. How did they actually prove it does 2 opposite things?
They utilized a really cool technique called in utero electroporation.
Oh, IUE.
Yeah, it's this highly precise method where you inject plasmid DNA into the fluid-filled ventricles of a developing mouse embryo's brain.
Well, it's still in the womb.
Right. And then you apply a rapid targeted electrical pulse. The electricity creates these transient, temporary pores in the cell membranes of the neural stem cells that line the ventricle.
And that just draws the custom DNA right inside the cell.
Exactly. And the plasmids they designed for this were genius. They created 2 modified versions of MAP1B.
Right, I remember this. The first version was fused with an NLS, a nuclear localization signal.
Yes, which is basically a chemical zip code that the cellular transport machinery recognizes. It forcibly drags the MAP1B protein into the nucleus.
And the second version?
The second was fused with an NES, a nuclear export signal, that physically kicks the protein out of the nucleus and traps it in the cytosol.
So they can force the protein to live exclusively in one location or the other. What happened?
The outcomes were striking. When they forced MAP1B into the nucleus using the NLS tag, the brain tissue drastically overproduced neural stem cells.
Because the nuclear MAP1B was constantly signaling the cells to maintain their stemness.
Exactly. They completely missed their developmental window to differentiate and migrate. The result was a massive accumulation of cells trapped deep below the cortical plate, entirely locked in a progenitor state.
Okay, I am still hung up on the biophysics of this, though, because MAP1B is a massive, bulky protein.
It's huge, yeah.
So in a natural, non-engineered state, how is it physically detaching from the microtubule network, floating through the dense cytosol, and squeezing through the highly selective nuclear pore complexes without an artificial NLS tag pulling it?
That's the million-dollar question. And the natural mechanism relies on a targeted post-translational modification, phosphorylation. Yeah. So MAP1B has a very specific microtubule binding domain, or MBD. Normally, this domain has a high electrochemical affinity for tubulin, keeping it securely anchored to the roads.
Right.
But specific kinase enzymes, and the researchers highlighted JNK1 and PINK1 here, they act as molecular switches. They attach a negatively charged phosphate group directly onto the MBD of MAP1B.
Oh, I see. It's like stamping a VIP pass onto the protein.
That's a perfect way to look at it.
Because that addition of negative charge alters the conformational shape of the binding domain. The affinity drops, MAP1B weakens its grip, and lets go of the microtubule road.
Exactly. And now that it is unbound, it's free to be recognized by important.
And that bouncers the nucleus.
Right, the bouncers. They shuttle it straight through the nuclear pores and into City Hall. And by utilizing phosphorylation, the cell has evolved this incredibly elegant efficiency.
Because that didn't have to build a new protein from scratch.
Exactly. Rather than transcribing and translating entirely new proteins to halt differentiation, the cell simply phosphorylates an existing structural protein, moves its location, and completely flips the cell's developmental fate.
That is so efficient. Okay, let's lock this phase of the mechanism in with a quick recap for you listening. Bullet 1. MAP1B has opposing functions based purely on its spatial location. Cytosol equals mature into a neuron. Nucleus equals stay a stem cell.
Bullet 2. Forcing MAP1B into the nucleus halts normal brain development, trapping cells in a stem-like state.
And bullet 3, phosphorylation acts as the mechanism, the VIP pass, to release MAP1B from the microtubule so it can actually enter the nucleus.
Perfect.
So why do you think biology evolved to use one single massive structural protein to control two entirely opposite cellular fates just by moving its location?
Well, it creates a perfectly synchronized feedback loop. Think about it. If the cell needs to build more structural scaffolding, it makes sense to pause genetic differentiation until the physical house is in order. The protein that builds the house is the same protein that tells the DNA to wait.
Wow. Okay, but if we connect this to the bigger picture, we have to look at what happens when this delicate spatial balance is destroyed.
Which takes us directly back to the clinical mystery. The patient's suffering from periventricular heterotopia.
Right. So to correct the pathology of PH, the researchers sequenced the genomes of 17 patients with the disorder.
And what did they find?
They found a series of de novo mutations directly in the MAP1B gene. And the most thoroughly studied one was a frameshift mutation called C.2133DLG.
Okay, let's break that down.
Yeah, so that is a deletion of a single guanine nucleotide. Because DNA is read in triplets, dropping just one letter shifts the entire reading frame.
Which is disastrous for a protein.
Totally. It results in a premature stop codon, which means the cell produces a physically truncated, shortened MAP1B protein.
And to observe this mutation in action, they didn't just rely on mouse models, right?
No, they went much more advanced. They used CRISPR-Cas9 to engineer this exact patient-specific frameshift mutation into human-induced pluripotent stem cells.
Wow, so human cells.
Yes, and they cultured those cells into 3D cortical organoids, basically miniature models of the developing human brain in a dish.
That's incredible.
Here's where it gets really interesting. When they used fluorescent tags to track the truncated MAP1B in these mutant organoids, they found something devastating. The shortened protein was entirely trapped inside the nucleus.
Wait, trapped? It couldn't get out?
It couldn't leave City Hall. It had lost the specific domains required to bind back onto the cytosolic microtubules.
Oh, wow.
And because it was hyper-concentrated in the nucleus, it was constantly blasting the signal to stay a stem cell. It severely delayed the differentiation of those progenitor cells.
And to quantify the exact downstream chaos caused by this nuclear trapping, the team employed single-cell RNA sequencing, or cRNA-seq.
Which is such a phenomenal tool.
It really is,
Yeah.
because it captures the active transcriptome, the exact mRNA being produced of thousands of individual cells simultaneously.
Right.
So when they projected the transcriptional profiles of the mutant organoids onto a computational plot, they didn't just see delayed stem cells. They identified an entirely novel, abnormal cluster of cells.
What did they call it?
They designated it the neurons unknown cluster.
Neurons unknown. That is, calling them unknown is so fitting because these cells were essentially suffering from a profound identity crisis.
Exactly. The sequencing revealed drastic downregulation in crucial migration genes. The most notable one was DAB1.
And DAB1 is essential, right?
Oh, absolutely. It's an intracellular adapter protein that basically acts as the GPS system for a migrating neuron. Without DAB1, the neuron has no idea which chemical signals to follow to actually reach the cortex.
So they're completely blind.
Blind and lost. And to prove these cells were actually lost, the researchers applied an advanced computational algorithm called RNA velocity.
Okay, I love this part. Explain RNA velocity for us.
So traditional RNA sequencing only gives you a static snapshot in time. But RNA velocity compares the ratio of unspliced pre-mRNA, so newly minted RNA, to fully spliced mature mRNA within a single cell.
Okay.
By calculating the time derivative of this splicing process, the algorithm can actually generate vector fields. These mathematically predict the future developmental trajectory of the cell.
Like tracking where it's going to go next.
Yes, and the RNA velocity vectors proved that the neurons unknown cells were migrating at incredibly slow velocities and taking highly tortuous, erratic paths, just completely curving and looping around.
If we connect this to the bigger picture, PH was previously classified purely as a migration disorder.
Right, we thought the roads were broken.
But this transcriptomic data proves PH is actually a fate disorder. The defect doesn't begin in the migrating neuron, it begins way earlier, in the stem cell stage. The trapped nuclear MAP1B rewrites the genetic identity of the cell so severely that by the time the neuron is born, it never even possessed the instructions for migration in the first place.
It's a fundamental paradigm shift in developmental neurology.
It really is. So let's do our third recap. Bullet 1. PH patient mutations cause truncated MAP1B proteins that get pathologically trapped in the nucleus.
Bullet 2. This nuclear trapping creates abnormal, confused neurons, which they call the neurons unknown cluster.
And bullet 3. pH is redefined. It's not just a problem with erodes, migration. It's a problem with the blueprint, cell fate.
Right.
Which makes you think, if a disease is categorized wrong for decades, how much does that delay us from finding the right cure?
I mean, the delay is unquantifiable. You cannot cure a disease of genetic fate by developing drugs that target cytoskeletal mobility. You're treating the wrong mechanism.
Exactly.
But to even begin designing a fate-modifying therapeutic, we have to drill down to the ultimate molecular mechanism.
Right, because we know mutant MAP1B is trapped in the nucleus causing chaos. But how does a structural protein actually alter genetic fates? What is its physical, hands-on mechanism inside City Hall?
To map those physical interactions, the researchers utilized co-immunoprecipitation, or co-IP.
That's where you use an antibody like a hook, right?
Exactly. You take an antibody specifically designed to latch onto MAP1B, drop it into the nuclear extract, and then pull it out to see what other proteins come up attached to the hook.
And what did the mass spec of that pull-down assay reveal?
It revealed that inside the nucleus, MAP1B directly binds to the BAF chromatin-remodeling complex.
Oh, wow. The BAF complex.
Yeah. And BAF is one of those critical epigenetic regulators in the human body.
Let's break down how that works for a second. Inside the nucleus, your DNA is tightly spooled around histone proteins to form condensed chromatin, right?
Right. And in that compacted state, genes are physically hidden from the transcription machinery. They can't be read.
So the BAF complex is this massive molecular machine powered by a motor subunit called BRG1.
Yes, BRG1. It uses ATP hydrolysis to physically slide those histones apart. It literally unwinds the DNA so specific genes can be turned on.
And the co-IP data prove that MAP1B directly binds to this BRG1 motor.
Exactly.
Yeah.
And the binding interface is astonishing. MAP1B grabs onto the BRG1 motor using its actin binding domain.
It's ABD, which immediately begs the question, is actin another classic structural road material? Is it in the nucleus too?
It is. The research has proved it's right there.
But proving that MAP1B, actin, and BRG1 are just floating in the same nuclear soup isn't enough, right?
No, you have to prove they interact.
Right, so to prove they physically form a complex directly on the DNA, they utilized a cutting-edge, super-resolution microscopy technique called DNA paint.
Which is a brilliant method for shattering the diffraction limit of light.
Because standard microscopes can't distinguish molecules that are closer than, what, 200 nanometers?
Exactly, 200 nanometers.
Yeah, okay.
But DNA paint uses these transiently binding fluorescent oligonucleotides. They constantly attach and detach from your target proteins,
Creating a sort of blinking effect.
Yes, a blinking effect. And by computationally analyzing the center of those blinks over thousands of frames, you can achieve sub-10 nanometer spatial resolution.
That is mind-blowing.
It is. And the DNA paint images were definitive. They visually proved that MAP1B, nuclear actin, and the BRG1 motor sit incredibly close to each other, physically tethered into a single functional unit anchored directly to the chromatin.
So what does this all mean?
Well, it brings us to the final piece of the pathological puzzle. What happens to this complex when MAP1B is mutated?
Okay.
To answer this, the team used a sequencing technique called cut and run. It stands for cleavage under targets and release, using nucleus.
Quite the acronym.
Yeah, but basically it maps the precise genomic footprints where a specific protein is sitting on the DNA.
And what did it show?
The cut and run data from the healthy organoids show that the BRG1 motor delicately binds to the genome, carefully balancing the expression of stem cell maintenance genes.
Okay, but in the pH mutant models, the ones with the truncated MAP1B.
In the mutants, the trapped, truncated MAP1B acts like a hyperactive clamp. It grabs hold of BRG1 and forces it to bind vastly more often and far too tightly to the DNA.
Oh, I see.
Specifically, it permanently props open the chromatin at genetic loci that control STEMBus. Genes like CCND2, which drives the cell cycle, and HMGB2.
So by permanently holding the chromatin open at those specific genes, the mutant MAP1B is just continuously flooding the cell with signals saying, stay a stem cell.
Exactly. It forces the delay in differentiation.
This raises an important question. If MAP1B is doing this, what about the rest of the cytoskeleton?
That is exactly what the researchers hint at. They explicitly highlight another structural protein called filament A, or FLNA.
Right, FLNA.
It's a massive actin-binding protein, and mutations in the FLNA gene are actually the single most common cause of periventricular heterotopia.
Which strongly implies that MAP1B isn't an anomaly. This direct mechanical to genetic communication loop is likely a universal mechanism of human brain development.
Before we conclude, let's lock in this final mechanistic sequence, bullet 1. MAP1B physically anchors to the DNA unwinding BAF complex utilizing nuclear actin.
Bullet 2. In patients with PH, the truncated MAP1B hyperactivates this chromatin remodeler, mechanically propping open the genes that maintain a stem cell state.
And bullet 3.
Oh.
This direct link from structural scaffolding to epigenetic regulation is the absolute root cause of the brain malformation.
Can you picture the physical scaffolding of a building actively turning the pages of the architect's blueprints? Because that is exactly what is happening in your brain.
It's a profound paradigm shift.
It really is. The cytoskeleton is no longer just inert steel and concrete. It is a highly dynamic messenger, a fate decider, and a master epigenetic regulator.
Beautifully said.
Thanks. I mean, we started this deep dive looking at a blurry, confusing clinical MRI, just a static pile of misplaced neurons, and we zoomed all the way down past the cellular city limits directly into the nucleus, down to a single truncated structural protein hijacking a DNA unwinding motor.
We've watched a disease be entirely rewritten from a simple traffic jam into a complex crisis of cellular identity. But, it leaves us with an incredibly provocative concept to consider. We now know that structural proteins like MAP1B and filament A are secretly doing double duty in the nucleus of our brainstem cells, actively regulating our genetic code.
Right.
So what are the structural proteins in our heart, our liver, or our immune cells doing to our DNA right this very second? Are we fundamentally misunderstanding the basic architecture of life across the entire human body?
Wow. It truly makes you wonder how many other cellular secrets are hiding in plain sight, just waiting for the next leap in transcriptomic or super-resolution technology to uncover them. Keep questioning the blueprints, keep looking deeper into the mechanisms of life, and thanks for joining us on this deep dive.